And thank you very much for the very kind introduction, or the kind invitation to be here to close this session and talk to you. I'm honoured to share the stage with some of the stellar scientists that have come before me, such as David Kelsell from the ESDR and Professor Oyama from the JSID. I'm very flattered, I was a little surprised, but I'm very happy to stand here for half an hour and talk to you about myself. It's one of my favourite topics. My remit was please discuss your experiences, your career, some of the technologies that you've used over the course of your scientific career, and any advice that you might have for people. So for the next half an hour, I will try and do that for you. I have two disclaimers to start with. The first one is this is a personal perspective. And I think anyone's journey in science is a personal journey. And I'll tell you how I got to where I am, but that doesn't mean to say that that's going to be a suitable path for you. As we heard from David, David has not travelled about much, as he well pointed out, within the M25. He's worked and lived in London. And I think arguably there's absolutely nothing wrong with that. Whereas on the other hand, maybe I've travelled around a bit and moved institutions, and maybe that's why I'm here talking to you now. The second disclaimer is just an industry affiliation, which has nothing to do with this talk, although I mentioned the company, so I should mention this disclosure. So the title of the talk is "It's the Data That Matters." So if I'm reading your paper, I don't care where you generated that data. I don't care who you worked with, and I don't care where you published it. I want to know the experiments that you designed and the data that those experiments generated and what it's telling us. After all, that's what it's all about, right? It's all about discovery and publishing your data and telling the community what you found out. And so I think that's super important, and it's the kind of driving thought process behind what I do. However, I'm not blind to the fact that these things are important. So I work at Thomas Jefferson University. It's a fantastic university. We're in a great department with fabulous faculty and colleagues, and it's run by our chairman, by a very, very famous individual, Yoni Wito. But maybe if that little box in the right-hand corner there said Harvard or Yale or Oxford or Cambridge, you might be taking me a little bit more seriously. Certainly, maybe if I had done my degree at one of those institutions, I would have been taken a little bit more seriously during the course of my career. It is about who you work with to some degree, and I'm going to talk about some of the people that I've worked with who've been mentors and had great influences on my career. And I've been fortunate enough to work with some super individuals and collaborate with some super individuals that really have had a major impact on where I am now. And of course, we all know that more people read Nature than they do the JID. And you want as many people to know about the work that you're doing, because the work that you're doing should be amazing in your eyes, right? I mean, that's why we do what we do. We do research, we make discoveries, and we should think that that's the best thing there is under the sun at that particular point in time. So you want to publish it in somewhere where other people are going to read it and appreciate it. So it does matter to some point where you publish. However, at the end of the day, when we've all lived full lives and passed on, it's the data that remains, and it's the data that matters. So publish your best quality data. And a bit of advice here, avoid predatory journals. Now, I guess everyone in this room who's ever published a paper or have been a corresponding author will get daily emails from some fake journal of dermatological research or something. And I in my career have published 2 papers. One of those papers, the words that were mentioned were normal heart development. And another paper, which was just a letter, mentioned cardiomyopathy. And pretty much every week I get maybe 5 or 6 emails from the Journal of Heart, Blood and Amazing Science. Please, eminent professor, submit your work. And it's just, I hit delete straight away. But, you know, as a young scientist, you get an invitation to submit a paper to a journal, and you think, oh, that's great. Yeah, I did publish a great paper last week. That's really nice. Maybe I'll look into it. Don't fall into that trap. My advice would be, if you don't know anyone on the editorial board, or if your colleagues or your chairman or your friends don't know anyone on that journal, just press delete. If you know someone on the editorial board, email them and say, you're on this editorial board of this fake journal of heart disposition. What's the paper, the journal all about? And if the reply is, oh, yeah, I accepted this invitation like 10 years ago. I haven't actually done any work on that journal, and yeah, I don't know what it's all about. Then perhaps the question is, why on earth are you on that editorial board? There was a great editorial in Nature just, I think, within the last month that said, you know, this is a major problem for us, and really the only way to stop the proliferation of these types of journals is to cut off the supply. Publish in the JID, or Clinical Experimental Dermatology, quality journals with people that you know working for them would be one piece of advice. The other piece of advice that I'll give you is work hard and work smart. These are obvious things. I tell my lab all the time to work hard and work smart. I have two little catchphrases. First one is papers don't write themselves. And again, I guess you should know this, right? It's pretty obvious. But unless you do the work, unless you do the reading, unless you do the writing, there isn't going to be a paper, right? And the other one is think before you speak, but read before you think. I think people just don't read enough, and I don't read enough, it's true, but you really need to read to be able to formulate those ideas. You need to know what's gone before. You have a fantastic idea. Someone had that idea 10 years ago and they published on it. If you don't read, you don't know about that. So really, this is my two pieces of advice. So I've broken this talk down to timelines, talking a little bit about technologies and my career, and then if I don't get too carried away and if I have some time, I just realized, oh no, I've set the timer. If I have some time, I'll talk a little bit about what's exciting in my lab at the moment. Hopefully that will happen. So I love a timeline. I love putting things on a timeline, and if someone asks me, oh, can you give a journal club about HRAS? I'll be there with my timeline. I'll have Howard Varmus discovering oncogenes. I'll have Alan Balmain there, and I'll have all the other papers afterwards because, yeah, it's good. It's good to put it up in a sort of context of time. So I graduated from Leeds in 1995 as a genetics graduate. I started my PhD pretty soon after that in London. It took me 4 years to get my PhD. I started my first postdoc. That took 3 years. I did the majority of a second postdoc, maybe 2.5 years, and then I started my first faculty position as a lecturer in London. Then I moved to Dundee, became a senior lecturer in 2011, and then in 2014 I moved to Philadelphia. Now I'm going to talk about this in a little more detail and acknowledge individuals that have been important in these moves and these positions, but as an acknowledgement to technology and the technology that's been important for my career, I've put down these little events on the timeline. So the first one is this, the ABI 310. This was the first commercial benchtop DNA sequencer that was available at the time. It was made by ABI. Capillary sequencing had been developed in the 80s based on, obviously, technology from Fred Sanger and developed by Leroy Hood and colleagues for capillary electrophoresis and fluorescent dye terminators. But really, it wasn't until 1995 where you could get a machine stuck in the lab, and a lab, my PhD lab was sort of 4 individuals, and there you could do your DNA sequencing. So that really was a landmark. And I started the PhD, and my boss bought one of the first ABI 310s that was available in small labs in London at that time. I guess the other thing was microarray technology. We've used this quite a lot. This was being developed by Pat Brown at Stanford at the time, and we were all super excited about that during the PhD. This is this time here. And then I started my postdoc, and there was retroviral gene therapy. This is the ex vivo bone marrow transplantation that cures patients with severe compromised immunodeficiency disorder. At that time as well, in 2001, we had the first draft of the human genome, and as Tamia said, I worked on the human genome, so that was pretty important. And then fast forward to 2005, where we have accessible massively parallel sequencing. We have the Illumina genome analyzer, and of course massively parallel sequencing was going on before that, but really this was the first commercial viable option to take your, get your genome sequenced, or certainly your exome. And then we have the first report of retroviral gene therapy for epidermolysis bullosa, which is the disease that I focus most of my efforts on. And I've just come from a meeting now to show that actually this has progressed even further, and please keep an eye out for the tabloids for that story, because it's absolutely amazing. And then we fast forward to here. Well, it depends on what you think is the first $1,000 genome, whether it's 2014 or 2015, but it's there, it's cheap, and actually BGI now will give you a whole genome at 30x coverage for $600, which is pretty darn good if you think that a human genome project, the initial draft of the human genome project cost $2.3 billion and took over 10 years to come to fruition. And then lastly, I just want to mention HSV1 gene therapy for EB. So in 2016, I was approached by a small biotech company who had a really great idea for gene delivery, particularly for collagen 7, which is a very large gene and has had problems with lentiviral and retroviral transduction because of the packaging. And they had this idea, they had this technology, and they said, could you do the preclinical data for us? So we set up a contract, and we looked at using this vector in patient cells, and we generated some really interesting data. So we go back, and I'll just go through the timeline and talk about the individuals that were super important in my career. So I joined Dan Nizetit's lab at the end of 1995. Dan was a technologist. He's Croatian. He did his MD in Zagreb, then he moved to Berlin to do his PhD, and then he joined Hans Lirak's lab at the ICRF in London as a senior postdoc. He then started his own lab in 1995, and I was his first PhD student. He's a technologist, or he still is a technologist. At the time, he was a technologist, and he was basically creating the libraries that facilitated physical mapping and the contigs, which David Kelso mentioned earlier. You're basically taking bits of human DNA, you're chopping it up, you're putting it into a vector, and you're then pulling out those pieces of cloned DNA and overlapping them to get a physical map of portions of the human genome. And it was a small lab. We had maybe 4, 5, 6 of us there working on different parts of the human genome. We were working on chromosome 21 and chromosome 1, and we contributed to 0.27% of the large-scale sequencing substrates which were sent to Jena in Germany and Cambridge, Hinxton in Cambridge, to the Sanger Centre, and they were used to sequence. And one of the things I'm most proud of is when I started my postdoc and the first draft of the human genome came out, I looked on the NCBI website, and there were some of the clones that I'd ordered sequenced, and you could find the sequence of those clones there in the first draft of the human genome. So I was very pleased about that. The first paper that I ever published was in the JID, and it describes this contig, 2.5 megabases, which covers the region on chromosome 1, 1q21, where there's a concentration of genes termed the epidermal differentiation complex by the late Dietmar Mischke. And this was kind of a turning point, really, in my career. I'd applied to a position in Dan's lab to look at leukemic transcripts in Down syndrome. He was a chromosome 21 guy. But the lab was really busy, and that's what their focus was. And I couldn't really carve out my own project, my own PhD from that. Luckily, Dan was part of this chromosome 1 consortium because he'd made a flow-sorted chromosome 1 cosmid library. And he said, well, I've got to do something for this. Why don't you take a look at making the map? And so that's what I did, and that's been very profitable. And it got me into skin, so I had to write a PhD thesis, and I had to have an opening introduction about skin. So then I had to learn about this fantastic organ. As we all know, it's the best organ. It's the largest organ. You have this proliferating renewable epidermis. You have stem cells. You have genetic transfer. And I got pretty excited about it. So then, I guess, serendipitously, I was looking to make a move out of molecular biology, and this guy here, as everyone I'm sure knows, Professor John McGrath, he just published in Nature Genetics identifying the causative gene for ectodermal dysplasia skin fragility syndrome as plakophilin-1, and he was advertising a position to look at the functional consequences of reintroducing plakophilin-1 into patient keratinocytes. And so that was my main project, and it was on skin, so I went and joined John's lab, and I have to say that probably was one of the best, if not the best, career move, because John is an extremely dynamic individual. He's very bright, he's very smart, he has such a great clinical eye for when it comes to genetic skin diseases, and I was able to help out with some of the projects that John was doing in the lab, and we were very productive during that period, and we identified, or John identified, the mutations in the causative in the genes for many genodermatosis during this time. And I learned a lot from John. John has always, and still remains, to put the patients first. So whenever you're doing your research, you put the patients first. I remember we just published in the Journal of Cell Science demonstrating some of the effects of re-expressing plakophilin-1 in patient keratinocytes. And John was like, right, so this is great, we've got the paper out, that's really nice. So tell me, what can you actually do for the patients? What does this tell you? What do you think? And, I was quite young at the time. I'm like, oh, God, I don't know. So you thought about it, and it made you think. And that's something that stayed with me for a very long time now in terms of you get up, what gets you out of bed in the morning? Well, it's discovery for sure, but it's translating those discoveries into patient benefit, and that's always stayed with me ever since. John also introduced me to this disease, epidermolysis bullosa, the high incidence of squamous cell carcinoma, and also the charity DEBRA, Deborah UK. John was instrumental in how Deborah developed and how their model of funding developed, because Deborah have been extremely insightful from those mid-90s onwards, have invested heavily in research. And research takes a long time, and I think we really are now seeing some of the benefits from that early, heavy investment in research. And John was very instrumental in that. But it wasn't, so we had a very successful time in John's lab, but it wasn't, science is not all about success. There were some not so successful encounters. So one of the reasons I joined John's lab is because I wanted to publish in Nature Genetics. I was a geneticist, right? That's the best journal in the field. And we tried, for sure. And on reflection, I don't know why some of those papers, which are now very highly cited, didn't get in, but they didn't. And they're editorial decisions, and you deal with it and you move on. So that didn't happen. The other thing as well is the very first grant application I was involved with, that John had written and I'd helped out with, we just identified the causative mutation of Kindler syndrome, identified mutations in KIND1, FERMT1. It was a component, it caused a variant of epidermolysis bullosa, it led to skin atrophy, it led to skin cancer. It was a component of focal adhesions, which in the field at the time was very novel. And we'd published, we'd identified the gene, we'd developed tools, we had cells, we had a vector, we had antibodies, and we put the grant into the Medical Research Council to understand what KIND1, FERMT1 did. And this grant was a winner, right? I mean, it's novel, you've got all the tools, it's very straightforward. We were dead sure it was going to get funded. And not only that, but we knew someone on the panel. And we were like, oh, it's going to get funded. But sadly, it didn't. And when we spoke to the guy on the panel, he was like, well, yeah, we're in the same institution, so I had to leave the room. I was told that we had, 20 applications, yours was ranked third, but we didn't have that much money this time, so we only funded 2 grants. And if anyone ever tells you that, I mean, they're just being nice, right? You didn't get the grant funded. That's fine. That's what happens. There's a whole number of reasons. Someone had a better grant. I think that's the best way to look at it. But that's going to happen, right? I mean, for me at the time, it was 3 months before the end of my first postdoc contract, and I really wanted to work on that grant, and we didn't have any money. So it was a little bit like, oh, right, well, that's kind of tricky. However, luckily, this guy had a postdoc position that he was advertised. He was actually working with John. Now, John, obviously, as I described, has been very instrumental in moving Deborah's scientific agenda forward. But John, he ran the tertiary referral clinics and still does, I guess, at St. John's and the affiliated organisations, and he sees a lot of patients. He's very heavily involved in the charity at that time. And the patients were telling John, look, we really need to know more about skin cancer, because skin cancer is the biggest cause of death in the patients with recessive dystrophic epidermolysis bullosa. And John had been really working hard on Ian Hart. Now, Ian Hart is probably a guy you don't know. He is a tumour biologist. He is actually a trained vet. He did his postdoc with Isaiah Fidler at the NIH and working on the B16 melanoma line. He then came back and started his own lab at the ICRF in London, and he did seminal work on integrins and tumour biology. He had a fantastic lab and a fantastic department, and John had been working on Ian to try and get into understanding skin cancer in the EB patients. And they had a grant. They had a grant to make a conditional knockout mouse of type 7 collagen. I really wanted to work on kindling. I didn't necessarily want to work on mice, but this was working in the field. It was a continuation. Ian had a fantastic lab. So I took the position, and it was a complete disaster, a complete failure. I didn't publish a single paper from this postdoc. I screened over 10,000 embryonic stem cells to try and get those homologous recombinants that we could then make the mice with. Didn't happen. I was doing a number of other projects. They weren't coming to fruition either. And the only person really I can blame is myself, but it taught me a huge amount, right? It was a great pleasure and a great benefit working in Ian's lab. He had some great people working with him, like Keraban Hodavala-Dilk, who'd done her PhD with Fiona Watt, had worked at MIT with Richard Hines, and also John Marshall, who was Ian's go-to man in the lab, who's now a professor and a very eminent scientist in the field of pancreatic cancer and tumour biology. So it wasn't a complete disaster, but it wasn't a success. And that's, we heard from David's talk about the peaks and troughs, and that was a real trough in my career. But luckily, about 2 years into this postdoc, and things weren't going well, but I'd worked in John's lab, so we were still publishing a lot of papers, and so I still look quite good on paper. This position came up. There was a lecturership being advertised in London in Irene Lee's department, working on understanding RDB-associated SCC, and that's what I was working on, and that's what I'd gotten interested in. And actually, underlying to that, this was really an initiative from John, because he wanted Deborah to invest in squamous cell carcinoma, and of course we all know Irene Lee's lab was a real focus of squamous cell carcinoma. So he really pushed Deborah to invest £1 million, the biggest grant that they'd invested before, on trying to understand EB-associated SCC, and part of that grant was a career development fellowship, and John suggested that I apply for that. So I did, and I got the position, and that was great. And that really has been the trajectory for my career. A year into working in Irene Lee's department, Irene said, "Oh, I just got a job in Dundee. I'm moving to Scotland. Would you like to come?" And now I'm an Englishman. I like football. And I don't know if anyone has ever watched football as an Englishman in Scotland or in the presence of a Scotsman. Now, I love Scotsmen, right? But they certainly hate the English national team. And I didn't really like the prospect of going all the way to Scotland and having to go to the pub and watch England play any other team. I mean, Diego Maradona for his hand of God goal at the 1986 Mexico World Cup. That man is a god in Scotland. He is an honorary Scotsman for scoring that goal and putting England out of the World Cup in 1986. And I really didn't want to go to Scotland, but my wife was quite amenable to it. She liked the idea. And essentially, Irene could offer me a tenured position. Now, there was opportunity to keep the grant in London, opportunity to keep it at the institute that I was already at, and I'd even spoken to John about possibly taking the grant to John's department because, yeah, I had fantastic productive time working in John's lab, and he is really a great guy. But nobody could offer me tenure apart from Dundee, and really, that's the other piece of advice. You've got to look after yourself. No one else will. I mean, you'll get advice. People will support you, there's no doubt. But at the end of the day, when the chips are down, you've got to think about yourself. So that was it. Right, I'm going to go to Dundee. So then I moved to Dundee. And really, it was a very good move as well, because you expose your research, and we generated some data in the 2 years of the start of that program grant. You expose your research to people with different views and different thought processes. I built up a relationship with John Forster, who was a really great clinical scientist who had a great perspective on microarray data, and we developed some really interesting findings in collaboration with John. And really, I think then the work took off, and we really made some great discoveries about RDB-SCC and defining the genetics and some therapeutic targets in there. I was made a senior lecturer in 2011, and at that time, things were going really well. I was a co-applicant on 3 program grants that totaled over £5 million. 2 of them were Irene's, and one of them was a guy, a breast cancer surgeon, Alistair Thompson. We just discovered that the microenvironment in EB was a very tumour-promoting environment, and we were applying some of the things we found in RDB to breast cancer. So I'd written some of these grants and written parts of these grants and contributed to them as a co-applicant. I had an industry contract. I had a couple of smaller grants. But I'd also started collaborating with guys in the US, and I was looking to the US at that point. I was collaborating with Raymond Cho at UCSF. I was collaborating with John Astor at Harvard. And I was really attracted by their dynamism and their dynamics. I was attracted by the dynamics and the dynamism of the United States. And I was really impressed by the structure of NCI-designated cancer centers, where they really spend effort to marry the clinicians and the basic scientists. Now, there are cancer centers in the UK. There's some great in Marsden, you've got the Beatson and other places, but really they're sort of few and far between, and they're very competitive to get into. And the US, that's the standard model. You have a very close relationship between the clinic and the science, and that was really attractive to me. At the same time, I'd been meeting folks from the dermatology department in Thomas Jefferson, and I give a shout out to May Mahoney. We met at an ESDR, salsa dancing in Helsinki at 1 in the morning. And we've struck up a great friendship since then. And I'd been talking to May, I really want to move to the US. She was like, oh, yeah, maybe you should. Actually, she dissuaded me to come. She was like, you don't want to come to the US. God, you've got it great in Europe. But nevertheless, I think she had a kind word with her chairman, Yoni Wito. And in 2013 at the IID in Edinburgh, I found myself in a bar late at night having a beer with Yoni Wito. And I said, Yoni, I really want to come to the States, work in a dermatology department that has affiliation with an NCI-designated cancer center. Yoni was like, let me see what I can do. And that was in May. And I heard again, I think I got an email from Yoni later on in the year, maybe September time, saying it's a possibility, we've got to sort out the budget. And then in February of 2014, we started to discuss the possibility of coming to Philadelphia. And in July, August, I moved out and joined the dermatology department there in Philadelphia, which has been a fantastic move. I've really enjoyed myself there. I am enjoying myself there. I certainly thank Yoni for investing in me, because a lot of my money, as I said, I was a co-applicant on grants. I had an industry contract, which actually moving an industry contract from the UK to the US turned out to be impossible. And I had very small amounts of money. So I had nothing to support myself. And as some of you well know, that you have to support your own salary in the US. So I'm very grateful for Yoni to take the chance on me to do that and support me for the time that I've been there. And it's been a good time. We've made some good translational discoveries. I mean, I received funding just last year to do the first trial of an experimental therapy in advanced patients, RDB patients with advanced SCC. And that experimental therapy has come purely out of the research that I'd been doing in Dundee prior. And so there's a really good example, taking John's advice, of really making translational research happen. And then, as I mentioned, I was approached by Crystal Biotech, who had this exciting new gene therapy vector. So they gave us the vector, we screened our patient cells and generated some really nice data. They used that data to raise money, and I was involved with some of the venture capital investment discussions about that data. I was involved with the pre-IND to the FDA, and Crystal Biotech just last week raised $50 million on the New York Stock Exchange, and they'll be taking their product into clinic early next year. And we were involved in that, and so I find that really satisfying, and it's been a great experience. Also, we've been making some progress in RDEB. We have a talk on Friday discussing some of the comprehensive genetic analysis using some of the technologies that I've developed, that we've been using over the years. Okay, and then I've got one slide. It's about the data. These are my publications, the number of publications in a year. And as you can see, I got a couple of publications out of my PhD in 1999 and 2000. And then I joined John's lab. We developed a lot of publications. And then I have a 2-year period where I didn't publish anything. And really, that's just, that's what happens in science. Sometimes things don't work out. Sometimes you make the wrong decisions. Yes, you can go back and check. If I could go back, I would do things a little differently. But that happens. So don't be too disparate if you take on a project, it doesn't work. Learn from those mistakes, as Chris told us in the last talk. And then you can see it's no surprise that the year with the biggest publications, I managed to convince Yoni to take me on in his department. So what's hot right now in my lab? Actually, I've probably only got 2 more minutes, so I'm just going to rattle through. 3 minutes. Okay, so I'm going to rattle through this very quickly. I have 2 very talented individuals in my laboratory in Velina Atanasova and Ignacia Fuentes. These 2 are very dedicated, very capable scientists. Velina is a PhD student who is joint PhD student with my chair, Yoni Wito. And Velina has a talk on Saturday at 10 o'clock, so please, I urge you to go along and see that talk. Ignacia is a PI for Deborah Chile, and she's joined the lab for a year to get some experience in cell culture. And I wanted to talk briefly about her work and her data, but so maybe I'll go through that very quickly. We're interested in the role of fibrosis and the development of squamous cell carcinoma, since our genetic analysis of fibroblasts in RDB really tell us it's those guys that are different, and those guys are driving the aggressive tumors. But fibrosis is pretty difficult to measure. How do you measure it? It's kind of increased tissue, increased stiffness. That's quite tricky to model. It's a gradual process, right? It's multi-system, and people will say you can't do it in culture. You can do it over time. But the animal models really aren't that great. It involves challenging the mice with damaging drugs. So we've been trying to work with primary fibroblasts, and that's what we do a lot in the lab. I work a little bit with mice, but that postdoc trying to make a conditional knockout mice put me off for life. I do a little bit, it's true. But we really focus on patient primary cells, and so we grow these cells to confluence. They secrete a lot of matrix. We detach the matrix, and then we remodel that matrix in suspension over time. It's shown here as well. And then we embed the matrix and we section it. This is trichrome blue. The red is the nuclei. The blue is the collagen. And you can see they look fairly similar. And you can see the extent of the sort of tissue remodeling over 7 days in suspension culture, which remodels the collagen, and we would advocate as a more physiological representation of the tissue. You don't see much difference at trichrome blue staining, but you do when you stain with picrocereus red, which binds collagen and is red under white light. But if you shine a polarized light on it, it exhibits birefringence, and that birefringence is an indication of the collagen thickness. So when you go from green to yellow to orange to red, you see a change in collagen thickness. And we can measure that, and we can compare. Clearly, these two images are different, and we can compare and measure that and show that in EB we have a continual, what we're calling collagen remodeling, but it's a continual expression of an increase in collagen, where here in the normal, the thin fibers, we have a decrease, and the red are mainly stable. And we can show this as pretty consistent pathology. In normal cells, we don't see a big change. These are 3 different pairs of normal. These are normal breast reduction fibroblasts. These are HN. These are from oral cancers. And you can see, we see in 2 of the 3 cases, a reduction in the thin fibrils. Whereas in EB, we see a consistent increase. And we also see a consistent increase in SCC. And that's our hypothesis, that it's this ever-changing remodeling extracellular matrix which is driving the cancer. And this is just a normal isogenic pair from a non-tumor part from the same patient as the tumor here. So we're using this model to determine mechanism and screen for drugs, and I'm going to wind up now. And this is the project of Ignacio Fuentes in the lab. I'm not going to talk about that. It's kind of just confirmation. It's Fourier transformed infrared microscopy, which is a kind of cool technique, but we haven't time to talk about that. And I just want to end with saying, don't take yourself too seriously. Now, I think that's been a theme from people here, speakers here, and you can't, right? Because rejection, not getting those grants, not getting the papers where you want them to get, that's part of being a scientist. But really, it's the data that matters. Do the best work you can and publish it and get it out there to the community. So this is actually from a couple of nights ago. It's a good friend of mine, Julio Salas Alanis. And this is obviously, as you well know, Yoni Wito. And sitting opposite taking the picture and making us laugh is John McGrath. So I have to acknowledge the funding agencies involved here. These have been very supportive and awarded grants and funding to keep me going while I'm in the US, and I'm very much appreciated to those. And then I have a huge list of people that have been influential in my career and helped throughout, and I haven't the time to go through everyone, and I've probably missed some people out. But thank you very much for staying to the end here and for your attention, and I'd be very happy to answer questions either now or later at the reception or the dinner. So thank you very much.